Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the deep dive. Today, we're doing more than just going through a textbook. We're aiming for, let's call it the ultimate intellectual shortcut.
A strategic deep dive.
Exactly. A strategic deep dive into the foundational science you absolutely need for competitive exams like UPSC. We're pulling together the most critical concepts from the NCERT Class 12 biology syllabus, focusing on things that, have a high linkage to previous year's questions.
That's right. And this isn't just about biology. We're looking at everything from molecular genetics all the way through to India's major environmental policies. The goal is to build that bridge between the pure science and its relevance for the general studies papers.
Right. Like understanding the lac Operon isn't just about some obscure bacterial gene.
Not at all. It's about understanding the core concept of cellular efficiency. And that idea has, parallels at the macro level, even in economics.
Okay, so before we jump into the genomic blueprint, we really need to take a moment to thank the people who make this kind of high-yield content happen. Simplified UPSC by PPP.
Absolutely. So here's our humble request. If this deep dive helps you in your preparation, please take a second to subscribe to the YouTube channel.
And hit that like button on the video, and please share a comment. Your feedback really does fuel everything we do here.
And a huge, huge shout out to all of our existing members. Your support is vital. It's what lets us keep this level of focused, comprehensive analysis going. And if you're in a position to help, please do consider joining the paid membership.
Every bit helps us maintain this quality. Thank you. Okay, so our mission today is pretty straightforward. We're going to unpack the key scientific breakthroughs and policy-relevant facts across a few big themes.
We've got the genomic blueprint, then science for food and health security.
And finally, India's crucial environmental policy milestones. Let's get into it.
All right, so we have to start with the most fundamental question of all. What actually is the genetic material? The history here is just a perfect lesson in scientific method. It all really kicks off with Frederick Griffith in 1928.
Oh, right, with the Streptococcus pneumonia experiments. That's the one where he mixed the heat-killed virulent S strain with the live, harmless R strain.
Exactly. And suddenly the harmless R strain became deadly. He called it a transforming principle. He knew something was passed from the dead bacteria to the live ones.
But the assumption back then was that it had to be a protein, right?
Precisely. I mean, proteins are so complex and diverse, they just seemed like the obvious candidate. The real proof, the definitive step, came much later.
With Avery, McLeod, and McCarty?
Yes, between 1933 and 1944. What they did was brilliant in its simplicity. They took the heat-killed S cells and methodically purified the different components, the proteins, the RNA, and the DNA.
And the key takeaway from their work wasn't just finding the answer, it was how they found it, the process of elimination.
It was all about the enzymes. That was the critical move. They added proteases, which chew up proteins, to the mix. Transformation still happened.
So not protein.
Then they added RNases, which destroy RNA. Transformation still happened.
Okay, so not RNA either.
But then, the game changer. They added DNase, the enzyme that digests DNA. And when they did that, transformation stopped.
And that was it.
Yeah.
That was the proof. DNA was the transforming substance, the genetic material.
That set the stage for understanding how DNA actually works, which, brings us to the central dogma. You have DNA making copies of itself, and then you have transcription,
which is copying the DNA code into RNA. And that follows the same basic rules of pairing, except, of course, adenosine pairs with uracil in RNA, not thymine.
Right. But the real jump in complexity, I think, is translation. That's when you go from the language of nucleotides in RNA to the language of amino acids to build a protein. There's no simple one-to-one pairing anymore.
It's a much more sophisticated code. And that level of complexity demands control. Let's talk about that classic example of genetic control. The lac Operon in E. coli. Why should someone preparing for a big exam really focus on this one bacterial system?
Because it's a masterclass in resource management. It's just beautifully efficient. The lac Operon is what's called the polycistronic system.
One promoter or one switch controlling multiple genes.
Exactly, which is very common in bacteria. So you have the I gene, which makes the inhibitor or repressor protein, and then you have three structural genes, Z, Y, and A.
Right. Z is for beta-galactosidase to break down lactose. Y is for permase to let it into the cell. And A is for transacetylase.
That's it. So by default, the cell is always making this I gene's repressor protein. And that protein physically sits on a part of the DNA called the operator, blocking RNA polymerase from doing its job.
So the cell is essentially saying, look, there's no lactose around, so I'm not going to waste energy building the tools to break it down.
Exactly. Energy conservation, that is the core insight here. But when lactose, the substrate, shows up, it acts as an inducer.
It flips the switch.
It physically binds to that repressor protein. That binding changes the repressor shape, making it fall off the operator. And boom, the path is clear. RNA polymerase gets access, and the cell starts churning out the enzymes it needs. It's a perfect on-demand system.
From that tiny gene to a massive undertaking. The Human Genome Project, the HGP, launched in 1990, calling it a mega project almost feels like an understatement.
Oh, absolutely. We're talking about mapping 3,164.7 million base pairs. The sheer scale is mind-boggling.
And it revealed some incredible things, like the average gene has about 3,000 bases, but then you have dystrophin, the largest known gene, with 2.4 million.
The range is enormous. But the most important strategic takeaway from the HGP isn't really the number of genes or bases. It's the understanding of genetic difference.
And that's where something like DNA fingerprinting becomes so relevant from a policy perspective.
Precisely. We are all, 99.9% genetically identical? But that tiny 0.1% difference is where all the variation is. DNA fingerprinting hones in on that, specifically on something called polymorphism.
Variations in repetitive DNA sequences, right? The so-called junk DNA.
Exactly, especially microsatellites. These are short repeating sequences that don't code for proteins, but they vary so much between individuals that they create a unique genetic signature.
So for an exam, the lesson is that the HGP gives us the raw data, and DNA fingerprinting is the application. It connects that data to forensics, to population studies, and to all the big ethical questions around genetic privacy that are coming up now.
Well said. Let's shift gears now from the code itself to how we apply this science to, feed ourselves. Food security, which in India means we have to talk about the Green Revolution.
And M.S. Swaminathan, the father of the Green Revolution in India, his work with Norman Borlaug was completely transformative. What was the core scientific innovation there?
It all came down to high-yielding semi-dwarf varieties. These plants could take up huge amounts of fertilizer and water and turn it into grain without falling over, a problem called lodging.
So this started with Mexican wheat varieties, Sonalica and Calliensona.
Yes, those were introduced in 1963. They were followed pretty quickly by semi-dwarf rice strains in 1966, like IR8 and Taichung Native 1. The result was a massive, massive jump in yields that really helped avoid famine.
A huge policy success, no doubt. But for a competitive exam, you also need the critical perspective. What were the downsides that we're dealing with today?
Well, the downside was the sheer resource intensity. These strains needed enormous amounts of irrigation and chemical fertilizers. Over time, that led to things like groundwater depletion and soil degradation. It was a necessary solution for a crisis, but it created long-term environmental trade-offs.
A classic case of a policy success creating a new sustainability challenge.
Which is where modern biotechnology now has to step in. The old way of plant breeding screening, selecting parents, cross-hybridizing, it was just so slow.
And biotechnology offers speed and precision.
Exactly. And the prime example is creating pest-resistant plants to cut down on chemical pesticide use. Let's talk about bin toxin.
Okay, walk us through the mechanism of that toxin from Bacillus thuringiensis. It seems a little strange that a bacteria makes a toxin that's harmless to us but deadly to an insect.
The genius is in its form. The bacterium produces the toxin as an inactive protoxin. It's a crystal. When an insect, like a bollworm, eats the plant tissue, the very alkaline pH of the insect's gut dissolves that crystal.
Ah, so the alkalinity is the trigger.
It is. It activates the toxin, which then binds to the insect's gut wall, punches holes in it, and the pest dies. Our stomachs, on the other hand, are highly acidic.
So the protoxin never gets activated in humans, it just passes right through.
And putting that gene into a crop like cotton completely changes the economics. It reduces the need for expensive, damaging chemical sprays, which is a direct benefit to farmer health and the environment.
It's a powerful story, and there's a similar one in medicine, right, with insulin production.
A very similar, powerful story. Before biotech, where did insulin for diabetics come from?
The pancreas of slaughtered pigs and cattle. And it worked, but it often caused allergic reactions.
Because it was a foreign protein. It wasn't quite identical to human insulin. Human insulin has two chains, an A chain and a B chain, linked by disulfide bridges. What Eli Lilly did in 1983 was revolutionary.
They use genetic engineering.
They used E. coli as tiny factories. They got one batch of bacteria to produce the A chain and another batch to produce the B chain. Then they extracted them and chemically joined them together.
The end result being
Structurally identical human insulin. Humulin. It completely eliminated the risk of allergic reactions and guaranteed a stable supply. It's a perfect example of solving a huge public health problem at the microbial level.
Okay, let's move to the final section. India's policy responses to environmental pressures. This is core general studies material, starting with air pollution.
Right. For big industrial polluters, you need technology. Things like electrostatic precipitators. They are incredibly effective. They can remove over 99% of particulate matter from exhaust fumes.
How do they work?
They use a strong electric field to give the tiny particles a charge. Then, those charged particles stick to grounded collection plates instead of going up the chimney. For gases, you use scrubbers, which often spray a mist of water or lime to capture things like sulfur dioxide.
But the real policy focus now has shifted to the invisible threat, hasn't it? PM 2.5. Why is that specific size of particulate matter so dangerous?
Because it's so small, 2.5 micrometers or less. That size allows it to get past all our body's natural defenses, deep into our lungs, causing serious long-term respiratory and cardiovascular damage.
And the challenge is that it comes from diffuse sources, not just one factory smokestack.
Exactly. Vehicles, construction dust. Which brings us to that landmark case study in Delhi, the mandated switch to compressed natural gas, or CNG.
Right, that wasn't voluntary. It came after a public interest litigation, a PIL, that resulted in a Supreme Court order. The entire public transport fleet had to switch by the end of 2002.
A massive, massive undertaking. And the policy logic was solid. CNG is better for three main reasons. One, it burns cleaner and more efficiently. Two, it's cheaper. And three, and this is a big one for reducing corruption, it's much, much harder to adulterate than petrol or diesel.
And that policy push continued with nationwide emission standards. We saw Farad Stage 4's MINRs rolled out for all vehicles in April 2017. The policy direction is clear. Regulate the source.
Okay, so from pollution control to biodiversity protection. Globally, the strategy often focuses on hotspots.
Regions with incredibly high species richness and endemism, but they're also under severe threat.
Right. There are 34 of these hotspots identified globally, and India is part of 3 of them. The Western Ghats in Sri Lanka, the Indo-Burma region, and the Himalaya.
And our main strategy is in-situ conservation, protecting species in their natural habitat.
We've built a pretty robust network. We have 14 biosphere reserves, 90 national parks, 448 wildlife sanctuaries. But what's really unique to India, I think, is the cultural tradition of conservation. Things like sacred groves.
Like in the Kazi and Gentia hills in Meghalaya, these are patches of forest that are considered sacred, where cutting any tree is forbidden. It's an ancient, community-driven form of a protected area.
And this idea of people's participation goes way back. We have to talk about the story of Amrita Devi Bishnoi. In 1731 in Jodhpur, she and hundreds of other Bishnoi literally sacrificed their lives, hugging kedri trees to protect them from the king's men.
An incredible story. And that act led the government to create the Amrita Devi Bishnoi Wildlife Protection Award.
And you see that same spirit decades later in the Chipko movement in 1974. Villagers in the Himalayas hugging trees to stop logging. It shows that the most effective policy often builds on these deep cultural values.
Let's touch on one last global issue. Ozone depletion. We're talking about the good ozone here, the layer up in the stratosphere that shields us.
And the villains of the story are chlorofluorocarbons, or CFCs.
So how do they do so much damage?
Well, CFCs are very stable down here, but when they drift up to the stratosphere, the intense UV radiation breaks them apart, releasing chlorine atoms. And a single chlorine atom acts as a catalyst. It can destroy thousands and thousands of ozone molecules before it's removed from the cycle.
The consequences being things like snow blindness, cataracts, higher rates of skin cancer.
Exactly. But this is also a success story for global policy. The Montreal Protocol, signed in 1987, was a landmark international agreement to phase out CFCs. And it worked.
So we've covered a huge range. We've gone from the tiny mechanism of a single gene, the I gene, deciding whether to metabolize lactose, all the way up to a Supreme Court mandate changing the fuel for an entire city's public transport system.
The common thread through all of it is regulation and optimization. Regulating the genome, regulating the food supply, regulating our impact on the environment.
And to wrap this all up, we have to acknowledge the immense value that's embedded in nature, which brings us to our final provocative thought.
It's a concept that links conservation directly to intellectual property. Biopiracy.
Biopiracy, using biological resources or traditional knowledge from a country or a community without proper authorization or, sharing the benefits.
The classic example is the attempt to patent certain varieties of basmati rice, a grain that was developed over centuries by Indian farmers. It just highlights this huge disconnect.
So the question we want to leave you with is this. Given the incredible commercial value of traditional knowledge and biodiversity, what practical steps, beyond just signing treaties, must nations take to truly safeguard this biological and intellectual legacy against exploitation in the coming decades, especially when it comes to compensation and benefit sharing?
Something to think about.
Indeed. If this deep dive was useful for streamlining your preparation, please make sure you hit the like button on the audio and drop a comment with your thoughts on that question. And if you can, please consider subscribing and joining the paid membership.
We really appreciate all our existing members. Your support is what allows us to keep bringing you these super useful deep dives. Good luck with your studies.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.